Sadly, every day in the UK, over 40 people die from liver disease. Some of these deaths could be prevented by having a liver transplant, but there simply aren't enough donors to keep up with the demand. In America, for example, only 30 to 35% who need a liver transplant ever get one, and in developing countries, it can be as low as 1 to 2%. But what if we could grow livers in a laboratory and use those for transplants instead? A team in Pittsburgh has now done just that in experimental animals. They've reprogrammed human skin cells to turn them into liver cells and grow mini livers in a dish. These were transplanted into rats, genetically modified to prevent them from rejecting the transplant. Eva Higginbotham heard from Alejandro Soto Gutierrez how they did it.
What we're trying to do is constructing new livers from people's skin cells. The first step is obtaining a cell from your skin. And then after that, we convert those adult cells into stem cells by forcing the expression of some genes that are called transcription factors. And once these cells are transformed into stem cells, we can differentiate them into different cell types of the liver. And once we have all those cell types established and functional, then what we did was we used donor rat livers. We stripped the cells out and we kept only the matrix of the organ. And then we regenerate that organ with human cells. So basically, you end up with mini human liver.
And what does the bioreactor look like?
These bioreactors basically look like a box where the organ is suspended. And after putting the cells in, then we can culture that organ and observe how that tissue start taking place, how cells start connecting each other. And with your own eyes, you can see the organ taking color and taking a texture of actual tissue. And then to actually test their functions, we look into their capacity to metabolize and secrete proteins just like a normal human liver will do. And then we transplanted these into rats. And these animals lived for about 4 to 5 days after the transplant. And we were happy to see that. It's a good beginning.
So the rats that you put the mini livers in, did you already remove their own livers?
First of all, we inhibit the capacity of the native rat liver to regenerate or to regrow. And just before the transplanting of these mini human livers, we take the same amount of cell mass. So we take about 30 to 40% of the liver out, and then we transplant. But instead of rat liver, it's actual human liver, so that we are able to maintain the rats with a little bit of rat liver and a little bit of human liver.
How do you know that the human liver was doing anything good inside the rat?
Correct. So basically the main vessel that goes into the native liver was deviated and connected to the new human liver. So there was very limited blood going to the native liver of these rats. So that's one reason. The second reason is survival of these animals. If you do the same procedure but without any transplant, these animals actually will have a very difficult time surviving even overnight. And the third one was that after the third and fourth day, we actually checked the blood of these animals looking for human proteins that are normally secreted by liver. And we found them. And that was the third way we were confident that these livers were working, at least for this limited time of 4 to 5 days.
So why did the rats die after 5 days?
Yeah, so we did an autopsy afterwards on these rats. And when we look into the human mini livers that were transplanted, we found that there were problems with the vasculature. We have thrombosis across the organ and in the main vessel that was connected to the human liver. So those are part of the challenges that we are working on to extend survival of these animals from 4 or 5 days to actual weeks or months. And those are the next steps we're taking.
Where do you see lab-grown human livers being used in a medical setting if everything comes together? So is the idea that, I get sick, I have a problem with my liver, so you're going to take some of my skin, grow me a new liver and stick it in me, and then I'll be set? Is that the plan?
That could be the plan, but the way we are visualizing this is that now genetic engineering and gene editing is so advanced that what we're trying to do is creating stem cells from a skin that are universal. So that's the way we see it, so that we can have human liver grafts available at any time for anybody that need it without having to take any kind of immunosuppressant.
An amazing step in the right direction in a very important area of medicine. Alejandro Soto Gutierrez, and that work has just been published in the journal Cell Reports.